Um, Thank you everyone for coming, and my name is Xueyi and I'm a PhD student, and today I will talk about my analysis of nanopore long-read RNA-seq data using short-read tools. And so, well, hide this. So first of all, I would like to briefly introduce short-read RNA-seq, that usually the RNAs are extracted from samples, and during the library preparation, they will be get fragmented and amplified. So, and after that, they will get sequenced. So what the raw data we get from the sequencer are the fragmented RNA sequences, which we call reads. And usually to do downstream analysis, people will map the reads to the reference genome so that people can know where of the genome does the read come from. And after that, they can do gene level counting so that they can get a gene level count matrix where the numbers means how many reads are mapped to each gene in each sample. And after getting that, people usually do differential gene expression analysis to know which gene are change their expression between different conditions. And currently, the most popular tools in bioconducting are Agile, Limma, and DRIMSeq. However, only focusing on gene level analysis is not enough, because in a lot of cases, a gene have multiple exons, and not all the exons are expressed all the time. So sometimes during the splicing of mRNA, some exons can be alternatively spliced out, so they can produce multiple isoforms from one gene. And in this case, if we want to do transcript level analysis using short-read RNA-seq, we will face some trouble. Although for some of the reads like this one, which is from an exon which is only expressed in one isoform, or for this one which contains the junction information between different exons, we can assign them to specific isoforms. But for most reads like this one, they can be mapped to more than one isoform, and it's relatively hard to do the analysis with short read. So we may seek into more suitable technology. And now we have nanopore sequencing, that the machine will record the electric signal when the RNA or DNA molecule goes through a nanopore. And with the electric signal, we can do base calling to get the sequence. But however, the sequence here we get are still containing the adapters and barcodes. So we need to do trim and demultiplex so that we can get the pure sequence, which is ready for downstream analysis. And a typical transcriptomic analysis of long-read RNA-seq looks like this, that although it's more beneficial to do transcript level using long-read, but since we already have the data, why not should we also try gene level analysis? So for the gene level analysis, it's quite similar to short read, that we map the reads to the genome and do quantification. And after that, we can do differential gene expression analysis. While for transcript level analysis, unlike short read, usually we have an extra step called isoform detection, so that we can know if there's any novel isoform in our dataset which is not annotated in the reference. And after that, we can map the reads to the isoforms which we detected. And also, if we want to skip the isoform detection, we can also directly map the reads to the reference transcriptome. And after the mapping, we'll do transcript level quantification. And for downstream differential analysis, we can either do differential transcript expression analysis, which is focused on the expression, or we can do differential transcript usage analysis, which focus on the proportion of different isoforms between different conditions. And to compare tools for long-read analysis, for transcript level analysis of long reads, we can use some spiking called sequins, that these artificial genes are located on an artificial in silico chromosome, and most of the artificial genes contain different isoforms, and those isoforms are already mixed into different proportions into mixture A and mixture B. And so in our dataset, we have two replicates from either a mixture A or a mixture B, and we use the nanopore PCR cDNA sequencing protocol on Gridan machine to do the sequencing. And for the mapping, we use Minimap tool, which was a tool just designed for long read, while for the quantification, we use feature counts function from the R-subread package, which can also be used for short read tool, for short read data. And here is how many reads we got from the sequence dataset. We can see that in average, each sample have a little bit more than 1 million reads. And for the and after that, we used the Agile to prepare the data format, and we used the limma-voom workflow, which was designed for short read to do the differential gene expression analysis. And here is the result. We can see that the result is quite good, that we only detected 2 false negative genes, while for the other genes, they are either true positive or true negative. And also we compared our log fold change estimated in the limma-voom workflow to the expected log fold change, which were calculated using the sequence annotation. And we see that the correlation is quite high, which shows that the estimation of log fold change is also accurate. And then let's move on to transcript level analysis. For isoform detection, we choose 3 tools: FLAMES, FLAIR, and TALEN. And these tools also contain the step of mapping and quantification. And for the differential transcript usage analysis, we choose the limma-diffSplice, DRIMSeq, as well as the combination of stage R to do stage-wise test. And before we move to the results, I would like to introduce the isoform structural categories. For example, here there's an isoform in the reference with known splice junctions, and for full splice match isoform, they can directly map to the reference, and all of the known splice junctions are present, while for incomplete splice match, they can also be mapped to the reference, but some of the known splice junctions were missing, while for novel in catalog or novel in not in catalog, there are some novel isoforms, but the difference is whether the splice junctions are known or they contain some novel splice junction. And also there are some other categories like antisense or genic or something. Then due to the limitation of time, I will not go into the details. And let's go to see the results, because our data is purely artificial, so it's only containing known isoforms. So there shouldn't be any novel isoforms detected, and the number of full splice match category should be, in theory, it should be equal to the number of transcripts in this in the annotation, which is marked as dashed line in the left panel. And while we look in, and we can see that FLAMES have the most full splice match and the least novel isoforms detected, so it outperformed the other tools. And also when we look into read counts, we can see that FLAMES have the most read counts also from the full splice match category. And since if we have too many fake novel isoform detected, then the downstream differential analysis will be meaningless. So we only use the output from FLAMES to do the downstream analysis. And we compared our quantification of sequence to the expected abundance also from the annotation file. And we see that the correlation is also quite good. And here is the results of differential transcript usage. We can see that for the genes or transcripts which should be DTU, they have detected to have a low adjusted p-value. And for most of the methods, the genes or transcripts that shouldn't be detailed or have a high adjusted p-value. And you may notice that the purple box here may look unusual, but because in this method, the true negative genes or transcripts are already filtered out. So in here, it's only showing 2 false positive genes, so it's not that bad as what it looks like. So we can say that all the methods performed well. So in summary, for the sequence nanopore data, the D-analysis using limma-voom workflow, which was designed for short read, showed good results. And for isoform detection, flames outperformed flare and talon. And all of the differential transcript usage analysis methods are performed very well on this dataset. And since we are now working on an artificial dataset, we're considering if we can also have a look into some real experimental data. So we have focused on our collaborators' favorite gene SmoochD1, and they have previously did a short-read RNA-seq experiment of SmoochD1 mutated and also wild type of mouse neural stem cell samples. And now we also did data sequencing, but we use the nanopore RNA direct cDNA sequencing with using all the same set of cells. And due to the time limitation, I will not go through the details, but we can I can say that although the sequencing depth is lower than usual shorter read RNA-seq, but the differential gene expression analysis results by using a limma-voom workflow is still comparable to the previous short-read experiment. And flames detected about 40,000 isoforms, and about half of them were novel. However, for the detailed analysis, we don't find any detailed gene which I think they might be true. And at last, I would like to thank my lab members and also Marnie Blewett lab and Mike Clark lab who contributed to the dataset. And also I'd like to thank Gordon Smith, Young, and Wei for giving me some suggestion to the analysis. And if you are interested in seeing more seeing my results, you may go to this link or scan this QR code to see our preprint of this work. So thank you very much.
Thanks so much, Xueyi. Are there any quick questions for Xueyi? Please put them in the chat box. There's one there. Thank you, Xueyi, for a great talk. I'm wondering, on average, what proportion of genes exhibit differential splicing and how much heterogeneity is observed in terms of the extent of differential splicing between single cells?
Yeah, that's an interesting question. Yeah, but I didn't have any look into single cell analysis, and but our lab member Louis has done some single cell analysis, long read analysis, and he may have some thoughts. And if you're interested in this question, you may look into his preprint.
Sounds like a question you can follow up on the chat. Oh, here's another one.
Yeah.
Flames outperforms the other tools in isoform detection when there is no novel isoforms present in the test dataset. I'm wondering how it performs with novel isoforms present in the test dataset.
That's an interesting question. Since for the other dataset, I mean the mouse dataset, because we still don't know whether the novel isoforms are real or not. So maybe we need more insight into this. And actually, we're considering doing some analysis that we can delete some of the annotation and the isoform from the annotation in the sequence dataset, and we can run the softwares to this on this dataset and using the isoform with some of the using the annotation with some of the isoform deleted, and we can see whether they can those isoform which should be there can be recovered by the method.
Thanks so much again, Xueyi, for a great talk.
Thank you.